Good afternoon, ma'am. Good afternoon, classmates. I am Janaya Di Bornalo, and I am going to discuss about the oxidation of lipids. Oxidation is the process where energy is produced by degradation of fatty acids. Oxidation of fatty acid takes place in mitochondria, where the various enzymes for fatty acid oxidation are present close to the enzymes of the electron transport chain. The most important theory of the oxidation of fatty acid is the beta symbol oxidation of fatty acid. Fatty acids can be oxidized by beta oxidation, alpha oxidation, and omega oxidation. These three fatty acids oxidation are only minor oxidation. So the first one is the beta oxidation of fatty acid. It is the breakdown of a fatty acid to AcetylCoenzyme A. AcetylCoenzyme A is actually a molecule that participates in many biochemical reactions in protein, carbohydrate, and lipid metabolism. Its main function is to deliver the acetyl group to the citric acid cycle. And if you don't know what fatty acids are, they are rich sources of energy. Beta oxidation is also defined as the oxidation and splitting of two carbon units at beta carbon atom. This results in sequential removal of two carbon fragments as acetylcoenzyme A until the complete oxidation of fatty acids. Beta oxidation occurs almost in all tissues except erythrocytes and adrenal medulla. Energy is released when fatty acid undergoes beta oxidation, and the beta carbon atom of fatty acid is oxidized. This oxidation of fatty acid is a cyclic process. It is a process that is strictly aerobic. The stages of beta oxidation. There are three major steps involved in beta oxidation. First is the activation of fatty acids. Fatty acids must first be converted to an active intermediate before they can be catabolized. As you can see in the picture, fatty acids are activated to acetylcoenzyme A by thiokinesis or AcetylCoenzyme A synthetase. The reaction occurs in two steps and requires ATP, coenzyme A, and magnesium. Fatty acid then reacts with ATP to form acyladenylate, which then combines with coenzyme A to produce acetylcoenzyme A. Two high-energy phosphates are utilized since ATP is converted to pyrophosphate. The enzyme inorganic pyrophosphorase hydrolyzes pyrophosphate to phosphate. The immediate elimination of pyrophosphate makes this reaction totally irreversible. The second one is the transport of acetylcoenzyme A into mitochondria. In this step, a specialized carnitine carrier system or the carnitine shuttle operates to transport activated fatty acids from cytosol to the mitochondria. This occurs in four steps. First, acyl group of acetylcoenzyme A is transferred to carnitine catalyzed by carnitine acyltransferasol. This is present on the outer surface of inner mitochondrial membrane. In the second step, the acylcarnitine is transported across the membrane to mitochondrial matrix by a specific carrier protein. And in the third step, the acyltransferase 2, it is found on the inner surface of the inner mitochondrial membrane, converts acylcarnitine to AcetylCoenzyme A. In the last step, the carnitine released returns to cytosol for reuse. The third stage is the beta oxidation proper in mitochondrial matrix. Each cycle of beta oxidation liberating a 2-carbon unit acetylcoenzyme A occurs in a sequence of 4 reactions. First is oxidation, second is hydration, third is oxidation, and the fourth one is cleavage. In oxidation, acetylcoenzyme A undergoes dehydrogenation. Dehydrogenation is the process by which hydrogen is removed from an organic compound to form a new chemical by an Fad-dependent flavoenzyme, acetylcoenzyme A dehydrogenase. A double bond is formed between alpha and beta carbons. The second reaction is the hydration. It is when enoyl coenzyme A hydratase brings about the hydration of the double bond to form beta-hydroxy acetylcoenzyme A. The third reaction, which is the oxidation, it is when beta-hydroxyacetylcoenzyme A dehydrogenase catalyzes the second oxidation and generates an ADH, or nicotinamide adenine dinucleotide. It is necessary for cellular development and energy production. It is essential to produce energy from food and is the principal carrier of electrons in the energy-producing process in the cells. The product that is formed is beta-ketoacetylcoenzyme A. In the cleavage, the final reaction in the beta oxidation is the liberation of a 2-carbon fragment, acetylcoenzyme A from acylcoenzyme A. This occurs by a thiolytic cleavage catalyzed by a beta-ketoacyl-CoA thiolase. The new acylcoenzyme A containing 2 carbons less than the original re-enters the beta oxidation cycle. The process continues till the fatty acid is completely oxidized. The oxidation of odd-chain fatty acids. Odd-chain fatty acids are less common. They are formed by some bacteria in the stomachs of ruminants and the human colon. Oxidation of odd-chain fatty acids is similar to that of even-chain fatty acids. At the end, 3-carbon unit propionyl coenzyme A is produced. Propionyl coenzyme A is converted into succinyl coenzyme A. The succinyl coenzyme A is an intermediate in TCA cycle, so propionyl coenzyme A is gluconeogenic. The alpha oxidation of fatty acids. This is the minor pathway for oxidation of fatty acids. This oxidation occurs at alpha carbon atom. It does not require coenzyme A and does not generate ATP. It occurs in human body, mainly in liver and brain, by removing 1 carbon from carboxyl end. The role of this alpha oxidation is to convert even chain fatty acid to odd chain fatty acid, which are the constituent of complex lipids in the brain. The process involves hydroxylation of the alpha carbon atom with a specific alpha hydroxylase that requires iron and vitamin C as cofactors. Alpha oxidation is the most suited for the oxidation phytanic acid produced from dietary phytol, a constituent of chlorophyll of plants. Phytanic acid is a significant constituent of milk lipids and animal fats. Rashads disease is a rare genetic disease that causes weakness or numbness of the hands and feet. It is rare but a severe neurological disorder. It is caused by a defect in the alpha oxidation due to deficiency of enzyme phytanic acid hydroxylase. Patients with this disease accumulate large quantities of an unusual fatty acid, phytanic acid derived from phytol, a constituent of chlorophyll. Several neurological symptoms of Rashads disease. First is polyneuropathy. It is the simultaneous malfunction of many peripheral nerves throughout the body. Another symptom is called retinitis pigmentosa. It is a group of rare genetic disorders that involve a breakdown and loss of cells in the retina. Nerve deafness, or it is sometimes called hearing loss. Last one is cerebellar ataxia. It is a loss of muscle coordination caused by disease or injury to the cerebellum.